This experimental method is used to reveal the properties of store-operated calcium entry, or SOCE, in cancer and skeletal muscle cells using fluorescence-based methods. This is achieved by recording the fluorescence of Fura2 in live cells, which is sensitive to intracellular calcium concentration. In addition, the fluorescence quenching rate by addition of manganese at calcium concentration insensitive wavelength is measured, which directly reflects the activation of SOCE. To study SOCE in structurally specialized skeletal muscle cells, muscles are dissected and skinned muscle fibers are prepared. The fibers are then treated with low-affinity calcium indicators, and their internal fluorescence is monitored. These methods demonstrate that SOCE exists in both non-excitable cells and skeletal muscles with graded activation and tight coupling to intracellular calcium store content.
The main advantage of this fluorescent-based technique over other methods like electrophysiology patch clamp is that it is convenient to use and can be applied to cell population study or high-throughput screening.
It is important to use the right excitation wavelengths for radiometric measurement and manganese quenching assay. So calibration of individual instrument is crucial because each instrument is different.
Visual demonstration of this method is critical as the mechanical skinning process of single muscle fibers is difficult to learn because it requires special hand-on training, in-depth understanding and learning.